Hello and welcome to Fast Medical Article Reviews. This is Mohammed. The article that we're going to be talking about today is called Upper Cortical Layer Driven Network Impairment in Schizophrenia. So what is this article is basically about that we have in schizophrenia patients, and specifically in the upper layers of the brain cortex, we see an increase in principal neurons and a decrease in GABAergic interneurons. That include the subtypes that express somatostatin, that express parvalbumin, that express the large cholinergic and carotenoid expressing cell, and small carotenoid expressing. And we also see a decrease in the VIPs. So, moreover, these two, the ones that express somatostatin and the carotenoid, are also decreased in the amygdala and in the striatum, respectively, other than the cortex. So it's not only in the cortex. And we can also see some transcriptomic genes changes, which include downregulation in energy metabolism and upregulation in the neurotransmission. So what we see here, this is a heat map comparing the difference in neurons and genes between controls and schizophrenia patients. So this is for the control and this is for schizophrenia patients. Again, this is for the control and this is for the schizophrenia patients. So as you can see here, if we compare, when we compare these two, the control, the schizophrenia, we can see that the illumination has actually increased in these parts in the schizophrenia. We see it here and we see it here. And those genes are markers that tell us that there were increasing principal neurons. And as you can see here, these markers are also shifted to the right, so they are higher in schizophrenia. On the other hand, we can see some illumination decreasing here and here, and these decreasing illumination are present in the gene markers for the GABAergic interneurons. So that means that there is a decrease in GABAergic interneurons. And we can see the shift in those genes also here. It's a shift to the left, and they are higher in control, less in schizophrenia. Here, in the top two slides, top two shapes or figures over here, we want to try to know how much change in gene expression should as normalized distance exist in schizophrenia patients. So these ones on the right, you can see they are higher, they are on the top of the other ones, and these two are higher on the, if you compare it to the other ones. So we would like also to know which layers in the brain cortex was affected most. So these L1 to L6 represent the brain cortex layers from up to bottom. So L1 is at the top, and L6 is the bottom. And you see the major changes. The heaviest colors are correspond to L1 and L2. Basically L2 and L3 the top layers. And we can see that the largest expression changes was in the GABAergic gene parvalbumin. Again, we have already mentioned that. And the biggest change in the principal neurons were in the L2-CUX LAMP5. And this is basic, and here is a higher resolution of this image, more details, and it shows that basically the same thing, that the changes in gene expression was highest in these 5 genes. Here in this slide, we want to see the inter-individual variability between control individuals and then compare it to the inter-individual variability in schizophrenia patients. So here you can see that the blue box plot, which resembles schizophrenia, is actually wider than the purple box plot represents control. So that means that schizophrenia patients showed significantly greater individual expression variability. And by the way, it's a trend that was pronounced across all neuronal subtypes. In this figure, when visualizing the distances between samples using multidimensional scaling, we found that while controls, the small purple circles were close together, the schizophrenia blue triangle samples were scattered in different directions. In the bottom figure, especially in keratin plus subtypes, we can see the the blue box plots, which resemble schizophrenia, are actually below the red box plots, which represents control. So this means that those genes exhibited robust downregulation in schizophrenia. Here in this slide, we want to see how the function of the affected cells, principal neurons and GABAergic interneurons, were actually changed in patients with schizophrenia. So, by the way, this dashed line represents the p-value cutoff value. So anything to the right is actually significant, and anything to the left is not. So this figure on the left represents the down-regulated functions, and this figure represents the up-regulated functions. So you can see in the down-regulated functions, the most changes, which are over here, are related to energy metabolism and protein biogenesis. You can see those long red long lines over there, but here it's short, so they are not. Here is not significant. On the shape to the right, which represents the upregulated functions, we can see that most change was related to the functions that include neurotransmissions and plasticity pathways. Here again, we would like to know how do the cell functions differ in schizophrenia patients. So the most negative enrichment, which correspond to the decrease in function and the heavy red color, we can see it here, here, and here. So these are the 3 points of intersection. So these two green dashed boxes are the intersection of these genes with these genes, and these purple box intersection is caused by the intersection of these genes. So the genes, the functions that was different were includes a hypoxia factor connected to impairments in brain morphology and abnormal nervous system development, and other genes are important for lipid biosynthesis and the antipsychotic response, and other genes are associated with structural brain abnormalities. So in schizophrenia, these functions were impaired and may correspond to the damage exhibited in schizophrenia patients. Thank you so much for watching. I'm going to try to leave a link in the description of the video. Thank you so much.